So as was pointed out, I'll be talking about some Bayesian optimization of adaptive enrichment designs. So we've had a little bit of a discussion about why you might use adaptive enrichment designs in the first place already today. So hopefully there's a little bit of motivation here. Also, I think a lot of the other topics we've covered today could fit within this framework. I've not necessarily gone into detail of many of them, but maybe we'll point out as we go along where some of these other topics might fit into the work we're about to discuss. So I'm going to start off by just giving a little bit of an overview of what I mean by adaptive enrichment trials, just so you know exactly what I'm thinking. We're then working in a confirmatory setting, so we're going to look at controlling the error rate with these designs before we do any kind of optimization. We can then talk about how to construct these Bayes optimal trials and simulate some results. So I don't have any real studies to talk about, but we can have a look at kind of what situations might these trials actually be useful in using our same decision framework. We're going to then go into some discussion about how you might go about designing adaptive enrichment trials, and I think this will tie into this discussion we've just had about how you actually implement some of these things in practice. Fair warning, I'm not going to have any conclusive remarks to make about designing adaptive enrichment trials. I'm more going to be highlighting points that you might need to think about as you go about the construction of them under the framework that I've looked at. We'll have a look at some extensions to survival settings, delayed responses and things like that, although not in the detail that we've just seen, and then we'll have some concluding remarks. Okay, so designing adaptive enrichment trials. For any of you who've seen me before, you'll recognise these guys. I never present alone. I get a bit nervous up on stage on my own, so I always bring some friends along with me. So I'd like you to imagine that this is our patient population for a phase 3 clinical trial. Now, the eagle-eyed among you will have noticed that we have some differences between our patients here. So let me just highlight those a little bit better for you. So in this case, we're assuming that we have some pre-identified subgroups for the clinical trial. I know that male and female is maybe a little less advanced than what you're using in industry. You'd be more likely using biomarkers, but this is a nice way to illustrate it for the purposes of the picture. Okay, so let's just split them up and think about what this means in terms of the treatment effect. We're going to have a control and a new treatment that we're comparing. So we're going to randomize our patients to treatment and control without worrying too much about the details of that randomization for today. We then have a treatment effect in each subpopulation and an overall treatment effect for the population as a whole. And from this, we can define null hypotheses that we're interested in for this particular trial. So for today, we're going to concentrate on a formulation where we have a null hypothesis in one subgroup of interest, and then a null hypothesis in the full population. We could have a null hypothesis in the complement of the subpopulation, or just two subpopulations that we believe behave differently under some biomarker, but we're not going to have a look at that for today, but the methods would apply in that case as well. Okay, so the question is, coming forward from phase 2, as we've seen already, we'll have a lot of uncertainty about what the treatment effect actually is. I'm going to categorize this by a prior distribution. This could have been elicited from expert opinion, or as we've seen, maybe from constructing estimates that have been adjusted for bias or what have you as you go through the development process. So the question is, who do we actually recruit for the trial based on our uncertainty at this point? So we saw in one of the previous talks that if you have two treatments or two populations to carry forward, you might struggle to decide which is the appropriate population to actually conduct the phase 3 trial in. So we could conduct it with everyone, focusing on the effect in the full population, or we could just conduct it in the subpopulation. These are the kind of the two standard options, the two fixed sampling alternatives that we'll be talking about today. But if you're really not certain which of these two to go for, this is where adaptive enrichment may be of use to you. Okay, so in an adaptive enrichment trial, we're going to split the patient recruitment into two stages. And in the first stage, we'll start by recruiting patients from the full population and getting estimates of the treatment effects that correspond to these patients that we've recruited. Again, randomization is present here, but we're going to start getting some messy pictures if I include that throughout the presentation. After stage 1, we'll conduct our interim analysis, and that's where we decide how to spend the remaining recruitment for the trial. Now, one important thing to note at this point, within what we're going to look at within this slide deck, we are assuming a fixed total recruitment for the trial. So we either recruit within the full population for the rest of the available patients, in which case we get our stage 2 estimates as we did for stage 1, or we spend all of the remaining recruitment in just the subpopulation and also therefore only estimate the treatment effect in that subpopulation. Okay, so we're going to go through some examples later on, so I just wanted to point out the distributional assumptions that I'm going to be making in those examples. The key point here is that we've got our fixed overall sample size, so we're just considering proportions of recruitment within each population, governing the distribution of the associated treatment effect. Okay, now as promised, within this setup, we want to make sure we control the family-wise error rate. So this is our primary concern in a confirmatory setting. We don't want to do the optimization without having guaranteed this strong control. Okay, so I think I've already said that. But the key issue here is that for each possible decision at the interim analysis, so for these two possible decisions here, we want to guarantee strong control of the family-wise error. This is going to free us up to make that decision in any way we choose. Now, we've seen some methods already today in a couple of the talks about how to control for these error rates. I've made some fairly simple assumptions here, but you could, of course, apply more complex testing methodology that's suitable for the situation, as long as that testing methodology allows this key point of ensuring that the control of the error is guaranteed no matter which decision you make at the interim analysis. Okay, so we're going to have an overall closed testing procedure today. So we're just going to find some way of getting to some combined p-values for the trial, and then rejecting if all of the tests that we're interested in are rejected for an individual hypothesis. So this is just the standard closed testing setup. We're actually going to use Symes method within each stage. Now, as I pointed out, we're looking at a subpopulation and a full population, so I think Cyrus pointed out nicely in his presentation that the Burnett method would potentially be preferable here because we have an inbuilt correlation, and we know that correlation from the fact that the patients belong to both the sub and the full population. But for today, we'll use Symes. It keeps the computations a little bit easier when it comes to simulation. And to combine stages, we're going to use the combination tests, again, that Cyrus talked about, and we're going to use the weighted inverse normal. I won't say any more about that, we've already seen that today. The overall testing procedure is a little different. So we're going to start off with the observations that we have from the trial. These are going to define our stage 1 p-values in the box up there. And then in stage 2, we'll have either p-values for both the sub and the full population, or we may have only obtained a p-value for the subpopulation had we enriched the trial. So in the first stage, it's simple enough. We can just apply Symes method or the Burnett method if we were wanting to, or any other example that you may think of. In stage 2, if we continued recruitment in the full population, so we have these two p-values, wrong button. We have these two p-values available, then again, this is just like stage 1, we can apply Symes method. Whereas if we only have recruitment in the subpopulation without the p-value in the full population, then the intersection p-value just becomes the p-value for the individual subpopulation. And then we combine the p-values across stages, as said before, with the weighted inverse normal. So this is our overall testing procedure. And the nice thing about this testing procedure is that, as I said, whichever option is chosen at the interim analysis, we have guaranteed strong control of the family-wise error rate. So this means that however we make the decision, we have that guaranteed control. So we can make the decision however we want. We could make it based on flipping a coin if we wanted. We could make it based on external factors to the trial, or as we're going to see today, we could use a Bayesian decision framework. So some people get nervous when you start talking about a Bayesian decision framework in this case, but hopefully I've convinced you that despite the word Bayesian, I've not impacted the frequentist properties of the trial. Okay, so we've had a little bit of a talk about gain functions. I'm going to be using quite a simplified one compared to the ones that we've looked at so far. This is just for illustrative purposes as to how you might use the gain function as an optimization tool. But we could consider utility functions with a cost and a benefit, as we saw in the previous presentation. The key is I want to break up the possible outcomes for the trial and assign some kind of gain or utility to each possible outcome. The nice thing about the gain is this then gives us a single measure for overall trial performance. So before we have a gain function, we have three possibilities of the way that the trial can perform. So this would be three probabilities that we might be looking at. And the gain function reduces this to a single measure of overall performance, which is going to be how we go about the optimization. Okay. So in the literature, within adaptive enrichment designs, it's probably worth pointing out that there are a couple of perspectives that the gain function has been constructed from. So there is the patient perspective, in which case, to simplify the work that's been done in this area, the gain function depends on the true treatment effects. So what benefit is this treatment actually bringing to the patients? Or the sponsor perspective, where the gain function now depends on the estimate that you have at the end of the trial. So essentially, how are you going to market this treatment after completing the phase 3 clinical trial? Okay. Whichever gain function we've used, the overall measure for performance of the trial is then given by the Bayes expected gain. So this is just integrating over that prior distribution we talked about before for the expected gain of the trial. So this is where our single measure of performance comes in. So if we have data from the first and second stage of the trial, we can rewrite this gain, this integral. Essentially, this is just a step on the way to optimization. So you can see that we've separated out the information from the first stage of the trial, which then allows us to optimize the interim decision given any particular observation for the first stage data. So we're going to choose to either carry on in the full population or continue in the subpopulation based on which choice maximizes the expected gain for the rest of the trial. So this is given a posterior distribution after the first stage data. Okay. So a little bit of discussion about how you might go about computing this gain. I'm going to talk about this from a simulation perspective because this allows us kind of a full choice in terms of how the trial is actually constructed. So if you were to step away from some of the more simple assumptions that I've made, this simulation may be necessary to find out the behavior of the rest of the trial. So we start off by drawing with, we start off by having a single observation or a set of observations for the first stage of the trial. Given that set of observations, we then are going to draw from the posterior distribution. And from the posterior distribution, we are then able to simulate corresponding values for the stage 2 data under each possible decision and estimate the gain fairly simply. Okay, so this is just now actually observed outcomes of the trial or simulated observed outcomes of the trial to estimate our gain. And as I said before, we then choose the action that maximizes this expected gain. Okay. We then want to estimate the overall performance of the trial. So the overall performance of the trial, again, we're going to go about this by simulation. The main difference here is that we now have to simulate the first stage of the trial as well. So we're now drawing from the prior and simulating a corresponding first stage of the trial. We then are going to evaluate the interim decision and simulate the second stage accordingly. The question at this point becomes, how can we evaluate this interim decision efficiently? So I'm going to show you one method that we used for the simulations to follow, but you may be able to think of other ways for your particular problem about how you might need to go about the simulation. Essentially, what we want to do is create a lookup table for this interim decision. So we want to pre-compute interim decisions in the region of the parameter space, and then apply these decisions when we are simulating trials to speed up the process so that we don't have to compute the exact correct decision with each iteration of the simulation. So to start off with, we're going to take 4 points in the kind of corners of our parameter space. So you may just think of this as some area of the parameter space that you're fairly sure the simulation is going to lie between. For values that drop outside of this on the lookup table, maybe you then individually compute the decisions, but if you choose this region big enough, that won't happen particularly often, so it won't impact largely on the computational performance. Okay, so each of these corners, we can now compute the Bayes optimal decision, where the true decision is given by the dashed line. The dashed line isn't actually any true decision, it's just something I drew to illustrate this example. At the next iteration, we want to then create the next level of grid. So essentially we're going to select the midpoints of the current grid, and we want to evaluate the decision at each of these points. So to kind of make the algorithm a little bit simpler, we're going to say we're assuming essentially monotonicity of the boundary, and we're going to assign the decision of the nearest neighbours to any point, provided the nearest neighbours have a matching decision. So here we have the 4 corners where they don't have a matching decision, and thus at these points where we've now got the question marks, we're going to evaluate what is the Bayes optimal decision, again by simulation. Okay, so by simulation we filled in the next layer of the grid, and at this point you can already see where the computational efficiency will come from, in that for this quarter of the grid we no longer need to compute the Bayes optimal decision. We're making an assumption from this point onwards that the decision will always be to continue in the full population. So as we continue bifurcating to get the grid to the scale that we want, we needn't continue to compute the decision in all areas of the grid. Okay. So let's move on to our simulation study and have a look at some performance of the adaptive enrichment trials. So some basic settings. So we're going to assume that the subpopulation is half of the overall population. We're going to assume that we conduct the interim analysis halfway through the trial. Essentially, this is an assumption about the ratio of the sample size and the variance to fit into the distributional assumptions we had earlier. And then we're going to assume this prior distribution, and you'll see why I've picked this particular prior distribution in a minute. It's a nice example. So a corresponding decision boundary for this particular choice of gain and prior distribution. Oh, sorry, I didn't point out the gain. So the gain here, we're using the societal or patient version of the gain. It saves me having to worry about the estimation, essentially. So we'll notice some things about this plot. So the line in the middle, the black line, is the actual decision boundary. The dashed lines are then where the decision is within 5% in terms of expected gain. So again, this was computed using the bifurcating grid, just defining we want to look at more than just the boundary of equality. And we can see some discontinuities here, and this is caused by Syme's method. So this is a swapping between where you're looking at 2 times the minimum of the p-values or the maximum of the p-values. So it's what's driving the intersection p-value at the interim analysis that causes this. The other thing to notice is that this isn't just a linear boundary. So in terms of using thresholds, which is the other way that you might consider going about adaptive enrichment designs, you can see that the thresholds are going to be suboptimal compared to the Bayes optimal boundary. Okay, and the Bayes optimal boundary is, we've shown that it's optimal for any particular realization of the first stage data. Okay, So the results for the trial that used this prior distribution and decision rule. So let's have a look at the fixed sampling trials first, so these two right-hand columns. In the fixed enrichment trial, we only get a probability of rejecting the null hypothesis in the subpopulation because we only recruited the subpopulation. If we conduct the trial in the full population, these probabilities are spread out between rejecting only the null hypothesis in the sub, only in the full, or rejecting both null hypotheses. And we can see that for this choice of prior, the gain is equal under each of these fixed sampling methods. So this comes back to the start of the talk, where I was saying under this prior distribution, you might not be sure which fixed sampling method is the right choice of trial design for your particular setting. So this is exactly what's happening here. The adaptive enrichment trial, we can then see we've got kind of a compromise going on in terms of the probabilities of rejection between the fixed enrichment and the full population trial. And this is because we're enriching the trial around 60% of the time. Now the one thing that you might be thinking is, well, actually this improvement in gain is not hugely impressive. And actually this turns out to be the case for adaptive enrichment. Although it is capable of outperforming the fixed sampling methods by providing that compromise, because of the nature of that compromise, we never get a massive jump in performance compared to the fixed sampling alternatives. Which is why we were interested in a case where we genuinely weren't sure what to do between the fixed sampling alternatives. Let's have a little bit of a delve further into why this behaves like this. So if we're now looking at particular choices for the parameter values, so just fixed parameter values or point priors. In this case, where there's actually a treatment effect in the sub and the complement, we can see that conducting the trial in the full population was the obvious thing to do. Okay, it's far outperforming the fixed enrichment trial, and the adaptive enrichment trial is actually a little bit behind for this particular choice. So this adaptive enrichment trial, just to point out, is still constructed using the gain optimized under the prior. So we can see that for this particular case, actually, you would choose to do a full population trial. If we have no effect in the complement, we get the reverse, in that the fixed enrichment trial becomes obviously the best choice. And the adaptive enrichment is again in second place, but making a reasonable compromise between the two. And one last example, somewhere in the middle, we're getting similar performance across all three trials. The nice thing is that the adaptive enrichment trial, because of this kind of average performance at many places in the parameter space, that's why it can outperform the fixed sampling trials when you're averaging across the right prior. Okay, so on average, it's performing reasonably well everywhere, whereas the fixed sampling trials are performing well in specific places. So let's have a look at priors now centered on a couple of specific places, just to hammer that point home. This is where the treatment effect is good in both the sub and the full population. And again, we see, even with the variability, the full population trial now becomes an obvious choice of trial. Likewise, if we switch to a case where the sub is particularly good, but the full population doesn't get much benefit, the fixed enrichment trial becomes the natural choice of trial. However, there's one other thing to note at this point. The fixed enrichment trial, if we're in a development program where we have people interested in the effect of this treatment in the full population, before conducting the trial, the fixed enrichment trial may be a difficult sell. It's not statistically, the numbers speak for themselves, but you may find it difficult to get buy-in from a development team to say, actually, we're just going to abandon the full population without getting any more data from them. In this case, we can see that the adaptive enrichment trial it's still performing reasonably well with comparison to the fixed enrichment trial, and it is still outperforming the full population trial for this case. So this might be a little extra motivation to use it. You know that it's not the optimal choice of trial, but you know it's not going to be far off if you can't get buy-in for the trial that is statistically better. And again, so we'll have a look at the reason for this over the whole parameter space now. you can see why. There's this small region where the fixed enrichment trial is a little bit better than the two fixed sampling trials. But then as we spread the probability, the adaptive enrichment trial very quickly starts being outperformed by the fixed sampling alternatives. Okay, so any prior that's giving you kind of anything up here, you would say, well, we really should be doing a fixed sample in the full population. Anything down here, really statistically, that's a fixed sample, a fixed enrichment trial. Whereas if you've got some uncertainty and you're kind of in this region, then an adaptive enrichment trial may be the better choice. So let's have a little discussion about actually constructing adaptive enrichment trials. So the decision framework that we defined earlier requires two main things. We have the prior distribution and the gain function. And prior elicitation, that's not something that I'm really going to go into here. This is a fairly well-established topic. The gain function, I was going to have a little bit of discussion about it, but I think we've seen better discussion than I can provide here in the previous example. But other things that you might incorporate into it that we've not necessarily thought about. So we had efficacy, but we haven't thought about things like safety and treatment effects and estimation or marketing information. These are other things that you could incorporate into the gain function. The question is, how do you build one that truly reflects what your thoughts are on the trial? What's the important things to put into the gain function? Likewise, as I mentioned, the hypothesis testing structure, we don't need to follow this particular hypothesis testing structure. So a really easy example is that we swap the ordering of when we apply the weighted inverse normal and when we apply Syme's method. But the key point, as I said before, is that we must just ensure this strong control under any possible decision that we want to look at during the interim analysis. So we can look at some of the methods discussed earlier, or choose your favourite method for hypothesis testing, and you can probably make it fit within this framework. The next thing to talk about is how do you actually sample size and recruit for these trials? There are three things to consider when talking about the sample size. The obvious one is, what is the actual sample size for the trial? This is the thing that we're all fairly familiar with. But there are a couple of other things that we can use to optimize the trial, in that we can optimize the timing of the interim analysis, and we can optimize the proportion of patients we recruit from the subpopulation. So these are things that you might need to change to get the trial performing how you want it to perform. In terms of how you want it to perform, we can look at a couple of things. Again, we can look at the probability of rejecting the null hypotheses. These are the operating characteristics that we've looked at previously. We can also look at the gain function. Are we satisfied with that level of gain? Again, this depends on how you've constructed it and what it means to you. Another thing to think about is, having designed a trial based on these things, how do you present that information back to the rest of the trial team? So you might be satisfied with the sample size from a statistical point of view, but if it's constructed on these complex things, how are you going to present that back to everyone else? Estimation for point estimates is relatively simple in that it's just essentially from the pooled data. But you must give consideration to how you're going to construct confidence intervals in these cases. We've used a non-trivial hypothesis testing structure that's combined across stages and across different populations, and so that is something that needs to be given more thought. Okay, let's move on to some extensions and try and wrap up from there. So the extension that I really want to talk about is the extension to delayed responses. So in stage 1 of the trial, we now have this set of patients that we've recruited and got observations from. And we also have a set of patients who we've recruited, so we can no longer influence what happens with them, but we don't have their observations at the interim analysis. Hypothesis testing in this case is still relatively easy. We now treat the stages as recruitment cohorts and we're essentially done. But there's going to be more uncertainty about the interim decision making. So we're going to have to incorporate that uncertainty. One way to improve this would be perhaps a joint model between a long-term endpoint and a short-term endpoint. Because this decision making doesn't impact the hypothesis testing in any way, this joint model will not impact the hypothesis testing, so this is purely about how we make the decision. So this may be an easier sell than trying to use joint modelling in your final hypothesis decision. Your final hypothesis can still be based on the long-term endpoint only at the end of the trial. I'm going to skip quickly through survival, but there are some parallels to survival in this case. And then we'll end up the discussion. So the key points for the adaptive enrichment trial in these cases are strong control of the family-wise error rate must be guaranteed by the hypothesis testing structure for each possible decision. The Bayes optimal rule is then optimal for all realizations of the first stage of the trial, so it is that by definition it is the optimal way to make that interim decision. And then our comparisons with fixed sampling alternatives have shown that actually Bayes optimal adaptive enrichment, when it can offer a benefit, you need to make sure that you're using it under the right circumstances. It's not a just cure-all kind of problem. It doesn't just cure all problems. It's not always wise to use an adaptive trial if there's an obvious solution from a fixed sampling standpoint. And we can see that the adaptive enrichment designs, as presented, can be quite flexible. So we talked about delayed endpoints, we talked about survival. I talked briefly at the start that we could add subpopulations to these trials, or we could add things like early stopping for futility. So I didn't do that here because I assumed a fixed sample size. But as soon as we add, we talked about the costs of sampling earlier, as soon as we add that kind of structure into the gain function, we can then motivate early stopping for futility, because there might be a benefit to not continuing to recruit patients if there is no benefit to them. Okay, and then there's my references for those that are interested. Thank you for listening.